So thank you very much for the opportunity. I would like to thank the organizers, of course, for the opportunity to present Capstan Therapeutics, a new company located here in San Diego, and also thank you for your attention. So Capstan is about leveraging fully the potential of immunotherapy through in vivo reprogramming of immune cells, as well as exploiting that approach for the purpose of reprogramming also hematopoietic stem cells. These are my disclosures. So I'm going to take you through several pieces of information describing our technology as well as its potential in just 10-15 minutes. We'll start with foundational science. Then we'll describe the platform technology, share with you some of the therapeutic areas we're interested in, as well as finish with our main focus, in vivo CAR therapy. First, let me describe a little bit the genesis of Capstan Therapeutics. You may recognize some of the names involved with this company. Our technology is at the intersection of three major areas that saw significant progress during the last 3 decades. First, chimeric antigen receptor technology with pioneers such as Carl June and Bruce Levine. Secondly, mRNA vaccines and therapeutics with pioneers such as Drew Weissman, also part of our scientific founding team. And major advances in the third area, nanotechnology, for the purpose of delivering various payloads, including mRNA. Our foundational science is coming from UPenn, and I would like to direct you to this paper that was published last year in Science. This particular work is a preclinical work that showcased the possibility of utilizing synthetic lipid nanoparticles to reprogram in vivo the immune system, generate CAR cells at sufficient levels to remove or ablate pathogenic cells in a model of fibrosis, cardiac fibrosis. So this type of work brought in spotlight 3 major aspects. Number one can generate CAR cells in vivo through utilizing platforms such as TLMP. Number 2, one can generate CAR cells through using messenger RNA instead of viral vectors such as retroviral vectors. And number 3, one can actually position chimeric antigen receptor intervention for treatment in non-oncology indications. I'm going to take you through a couple of slides describing this foundational science. Number 1, what you see in this slide is the possibility to use a targeted lipid nanoparticle decorated with anti-CD5 monoclonal antibody, thus reprogramming all T cells in vitro as well as in vivo. In vitro on the left-hand part of the slide, showing the possibility to reprogram T cells expressing a chimeric antigen receptor against fibroblast activated protein used in this model. And on the right-hand part of the slide, reprogramming T cells directly in vivo with quite a significant number of T cells that are engineered. In vivo, CAR engineering is not compatible nor relies on lymphodepletion conditioning. So the total number of CAR T cells that can be achieved through using an optimized TLMP platform technology could be quite high. Our collaborators showed that they can produce in vivo chimeric antigen receptor T cells against fibroblast activated protein at a level with sufficient persistence also to ablate pathogenic cells, in this case activated fibroblasts in this cardiac fibrosis model. And this concept is being pursued by Capstan across a broader range of disease indications. In order for us to fully tap into this technology and move the field ahead, we had to rethink, redesign, and optimize the lipid nanoparticle technology. And this was done at 3 levels. Number 1, lipid composition, especially the ionizable lipid, which is important for the performance of engineering. We needed to invent new ionizable lipids that have increased biodegradability, allowing redosing, as well as decreased reactogenicity, compatible with higher dose of mRNA that needs to be delivered in order to reprogram the necessary number of T cells in vivo. Secondly, we needed to rethink the payload architecture, both the chimeric antigen receptor sequence as well as the mRNA optimizations, in order for us to achieve that level of performance in terms of expression and area under the curve needed to address therapeutic indications with variable and even higher therapeutic efficacy bars. And number three, we needed to actually optimize the platform technology to enhance selectivity at cell level. For example, if this platform technology is applied to generating CAR T cells in vivo, we needed to decorate the particles with binders that are able to increase the uptake by subsets of interest, such as immune effector cells. Okay. So this is how it works. On the left-hand part of the slide, we describe the concept of reprogramming T cells in vivo, utilizing targeted lipid nanoparticles. And on the right-hand part of the slide, I described the concept of delivering more complicated gene editing systems or payloads in order to correct or compensate for genetic defects. So these targeted lipid nanoparticles are rapidly taken up within minutes, hours maximum, by target cells, in this particular case, T cells, in a receptor binder mediated fashion. They are actually internalized in the endolysosomal compartment, where the ionizable lipid induces a breach in the endolysosomal membrane and allows the transfer of the payload in the cytoplasm. That's where actually the translational process occurs, and the chimeric antigen receptors are translated rapidly from mRNA, expressed on the T cell, and redirect rapidly the T cells in the microenvironment or remote tissues where the antigen is located to ablate pathogenic cells, be those tumor cells or non-tumoral cells. In contrast, of course, owing to the multiplexing capability of these targeted lipid nanoparticles, the delivery of gene editing systems requires, of course, the internalization of these particular components into the nucleus, where the gene correction is induced or the compensatory changes are elicited. Our interest is broad, and this is supported by the scalability, tunability, redosability of our targeted lipid nanoparticles. So in addition to B-cell-related diseases, both in autoimmunity area as well as oncology, we're interested in fibrosis, solid tumors, as well as in blood monogenic disorders such as sickle cell disease or beta thalassemia. We strongly believe that the future of CAR T-cell intervention is in vivo, and this is due to multiple factors. Number one, access. As we know, with all the advances during the last decade that we've been involved with, there are tremendous access limitations for products out there for B-cell malignancies. And utilizing a platform technology that is devoid of cells, devoid of viral vectors, devoid of viral components, that is scalable, tunable, would greatly facilitate access to CAR T-cell intervention. But it's not only about access, it is also about clinical performance. So utilizing a tunable approach, a synthetic platform technology, would allow us to meet higher therapeutic safety bars in disease indications that are very large, such as autoimmunity, regenerative medicine, and fibrosis. That's not something we can easily do with a cell-based or viral-based approach. At last but not least, efficacy. we are not relying on lymphodepletion conditioning. We do not interfere with the capability of the endogenous immune system. Hence, we have the opportunity to build on it mechanistically and through multiplexing and aspire to treat or tackle therapeutic areas with very high efficacy bars, such as solid tumors. And this is how it works in contrast with ex vivo engineered CAR T cells. So the driving force behind ex vivo viral engineered CAR T cells are juvenile stem cell-like memory cells in the infusion bag that activate and expand very rapidly post-infusion in patients who needed to be lymphodepleted or conditioned in order to create what we call immunological space. And this process cannot be controlled very easily because we have, in context of this technology, integrating viral vectors, right? So these products are associated in a subset of patients with serious adverse events such as ICANS, neurotoxicity, CRS, cytokine release syndrome. In sharp contrast, in vivo reprogramming of the immune system and generation of CARs in vivo does not rely on these elements, but relies on pre-existing effector cells in the body. So we are directly engineering those in secondary lymphoid organs where the T cells are mostly located. So those chimeric antigen receptor cells that are transiently reprogrammed with mRNA have the capability to engage very rapidly pathogenic cells and eliminate those. And this is a schematic diagram of how this would work in context of our main feature of our technology, which is the transient nature of CAR expression. And the key goal, key objective that we would like to achieve is true transient rational reprogramming of the immune system to generate durable responses, both in oncology as well as non-oncology indications. Perhaps many of you are familiar with the tantalizing accumulating number of reports during the last couple of years on how chimeric antigen receptor technology in its conventional format, ex vivo engineered cells, is potentially applicable even to autoimmune disorders, not only cancer. So we have accumulating evidence across 3 major indications in autoimmunity: systemic sclerosis, systemic lupus erythematosus, and myositis, that CD19 CAR T-cell intervention may be able to achieve the holy grail in this particular disease category, which is the immunological reset, namely through transient CAR T-cell activity, deep depletion of memory autoreactive B cells in this case, allowing then recovery of the normal B cell lineage without disease relapse. That's exactly what we would like to achieve, but using an in vivo synthetic platform technology that would obviate issues related to access as well as clinical performance, especially toxicity, which is very difficult to avoid with this conventional ex vivo CAR T-cell intervention relying on viral vectors. We have a vast data set that we generated during the last 10, 15 years in context of development of products such as Yescarta and Kymriah. And this data set points to performance metrics that we need to achieve with our in vivo technology in order to generate potent and safe product candidates that are transformative.
Thank you for your time, Mr. Bott. We are at time at this moment.
Thank you very much. And I'm going to leave you with this particular slide, again, with the conviction that through an in vivo reprogramming strategy, utilizing a scalable and synthetic platform technology, we can tackle larger disease indications in a safer fashion. Thank you very much for the attention.